record. Okay, so any questions about grade stuff before we move on? Any questions about what I have in the grade book? The thing that I have about the Blackboard time, that's just letting y'all know that I could kind of see where everybody's at. And then I found out later that I'm not sure that Blackboard's counting the time that you're spending watching lectures. I can't tell. It should be, but I don't think it is. So I put that there just so that you can kind of be like, okay, am I spending enough time or not? That's not affiliated with the grade. Everything else is. So everybody clear on all that stuff? All right, let's talk about chapters 12 and 13. Okay, so let's kind of think about where we were last week. Last week, we were talking about cell signaling. So I told you guys it's kind of a weird outlier chapter, but just to recap what you need to know from that chapter, we were talking about how individual cells start to stack up to become tissues, and how can you stitch them together? So we had a series of structures that y'all need to know that function to stitch cells together, all right? Then the second topic of discussion was, once you've stitched those guys together into tissues, how can they begin to communicate with each other? And we had two categories of communication, right? So we had next door cells and how they communicate, which is really just by building tunnels between each other. And then there were distant cells and how they communicate, which was when we talked about the different kind of hormones, okay? So that's where we were last week. This week we're, excuse me, really switching gears, okay? Still talking about cells, still talking about cell function, but now we're kind of going back to individual cells again. We're going to be looking at how cells make more of themselves, all right? And so chapter 12 covers the process of what we call the cell cycle, okay? And this is going to include the time that the cell spends just kind of hanging out and doing its cellular functions. Some of y'all might know that as interphase. Did y'all learn that like in high school, that term? Yeah. And then, so that's part of the cell cycle. And then there's a part of the cell's life cycle, if you will, kind of like the life cycle of a butterfly. So there's part of the cell's life cycle where it spends replicating, and that is mitosis, okay? So that's the subject of chapter 12. Now let's look at chapter 13. Chapter 13 is on a related topic. It also has to do with cell replication, but it has to do with a special case of cell replication called meiosis. Now, the thing that I find students get most hung up on, lots of times they know what they're talking about and they say the wrong word. So real quick, we're going to go over this in more detail, but if I had to ask you right here at the top of the lecture, can anyone give me a basic difference between meiosis and mitosis? What you got, Jennifer?
Meiosis is related to reproductive cells, and mitosis is related to the other cells.
Okay, when you say reproductive cells, is there a term that you could use?
Sperm and egg.
Okay, and is there a term that encompasses both sperm and egg?
Reproduction.
Gametes, that's what I was, yeah, I knew you knew it. I was just trying to get you there. Okay, gametes. So that is exactly right. Gametes is the term that refers to your reproductive cells, which would be your egg or ova and sperm, right? And so I'm going to tell you my 12-year-old, middle school boy humor way of remembering this. So meiosis has to do with sex. Meiosis. Okay, that's how I always remembered it in high school, like, oh, okay. I know you guys think it's dumb, and it is dumb, but you probably won't forget, okay? So yes, and if we wanted to look at this a little more closely, let's take a typical human cell, okay? So here's a human cell. I'm gonna draw this one here, and I'm gonna draw this one here. So to illustrate the difference between mitosis, which I'm going to put CHA, and meiosis, and this is kind of just a roadmap for where we're going in this lecture. To illustrate the difference, the first thing we need to know is how much genetic material we have in a regular human cell, because really the focus of this cellular replication is going to be copying the genetic material, because that's your instruction book. So you want to make a copy of your instruction book and distribute it among the copies so that everybody can function properly, right? So when I say how much genetic material is in the average human cell, what I mean really is how many chromosomes does the average human cell have? Wait, so who said that? What, how many did you say?
I'm not sure.
46? That's exactly right. Bada bing. And the reason I made you clarify is because lots of students want to tell me 23. Who wanted to say 23? Yeah, okay, you're not wrong. You're not wrong. It's just, well, you'll see how you're not wrong, Austin. Hang tight, okay? So in mitosis, what you're gonna end up with is you're gonna end up with 2 cells at the end of the process. And each of them are also going to have 46 chromosomes, okay? So yay, that's great. Now, when we say 46, Austin, the reason you're not wrong is because you have 23 different kinds, but 2 copies of each, one from mama and one from daddy, right? So that's why students often want to tell me 23. The difference has to do with what we call ploidy. Okay, so ploidy comes in 2 flavors. Actually, it comes in more than 2 flavors, but the 2 that we're going to focus on today are diploid. Okay, so diploid, di, that means 2. That means you have 2 copies of each chromosome, and the way that you can abbreviate that is 2N. But if you are a sexually reproducing organism, that means that there's a fertilization event that occurs where the gamete of one individual fuses with the gamete of another individual to make a third offspring individual. Y'all with me? So if you're a diploid organism, you are now presented with a very interesting problem. If I have 46 chromosomes and I meet my mate and he has 46 chromosomes and we decide to reproduce, which is a decision you really should think hard about. Just saying. Okay, so we decide to reproduce. If I take a 46 chromosome cell and he takes 46 chromosome cell and they fuse, suddenly I have a weird 92 chromosome baby, right? My babies are weird, but they ain't that weird. And then if my weird 92 chromosome baby finds a 92 chromosome mate, then you're up to what, 184 chromosomes? Yeah, that is crazy. So what we have to do if we are a diploid organism and we are sexually reproducing is we have to like reduce our number down. So that's why that 23 number is important, Austin. There's 23 unique different kinds of chromosomes. So what meiosis does is you go through 2 different divisions. You go through one where you go from 46 to 23. And you'll see why you actually have to do this division twice, because I'm going to go through that for y'all. At the end of meiosis, you started out with 1 and you end up with 4, okay? So meiosis it generates what we call haploid for like half. Haploid cells, which we can abbreviate as N, meaning that they just have 1 copy of each type of chromosome in them, okay? So that's where we're going in this lecture. All right, let's start out. First of all, we're gonna kind of focus on this idea of the cell cycle and mitosis, because that's the subject of chapter 12. Any questions before we move on to that? All right. So chapter 12 is cell cycle stuff. And where we're headed is we're going to mitosis. Now, in thinking about the cell cycle, I want you guys to pretend like we're in the 4th grade and we're studying like the life cycle of a frog or the life cycle of a butterfly. Y'all remember those little circular diagrams that you would get for those kind of life cycles? Let's draw one for a cell. So let's start out, and we have a little bitty baby newborn cell. And the first thing that's going to happen during the little baby newborn cell's life is it's going to undergo a period of growth, and it's going to undergo a period of time where it's just hanging out, doing whatever its cellular job is. If it's a liver cell, maybe it's detoxifying things. If it's a brain cell, it's receiving signals. So this first period is called the G1 period. That actually stands for first gap. Don't worry about that. But you can think of it as the first growth period. So in this phase, what we have here is growth, and then whatever the function of that cell is. Okay. Now, how long a cell spends in G1 really depends on the kind of cell that you're talking about. So for example, if we were talking about the cells that line the inside of your cheeks, those are turning over all the time. They're multiplying all the time because as you chew or brush your teeth or eat a piece of pizza, whatever, the interior lining of your cheek is constantly sloughing off and has to be replaced all the time. So those guys don't spend a lot of time in G1 because they're going to move out of G1 and into their replication pretty quick. So they're doing that pretty quick. On the other hand, you have things like, say, I don't know, bone cells, they're slower growing. And then you have some cells that actually get stuck in G1. And in your book, I don't talk about this much, but if a cell gets stuck in G1 and never moves out of that, it's sometimes said to go into a G sub 0 category. That would be things like your brain cells. Y'all know that when you're growing, your neurological tissue divides, right? And you end up with a set of brain cells that I don't know exactly when it is in growth and development, but there's a point at which you stop making more brain cells. And that's why traumatic brain injuries or spine injuries are so devastating, because if you injure your brain, those cells don't have the ability to divide and regenerate. That tissue just does not fix itself. Another example of cells that do this are fat cells. Did y'all know that you lay down your fat cells in childhood, and then as you gain and lose weight throughout the rest of your life, it's just that those cells are gaining and losing fat storage, but the number of cells stays the same. That's why childhood obesity is such a concern, because if you become obese early in life, you lay down more fat cells and therefore have a tendency to remain obese longer throughout your lifetime. Does that make sense to you guys? Yeah. So like me, I've been gaining weight like crazy over the last couple of years. So my fat cells, and I used to be a skinny kid. So I imagine I got some big old honking fat cells at this point in my lifetime, okay? So it just depends that the duration of this G1 phase depends on the type of cell you are. But after a period of time, the cell will reach a phase where it generates some cell signals that get talked about in your lecture. This is called a checkpoint here. And if the cell generates some signals, then it's going to move into what is called an S phase. Now, this S stands for the word synthesis. Synthesis, there we go. Anybody know what we're synthesizing or making during this phase? Copying something, we're making more of something in this phase. DNA, yep, you're synthesizing DNA. So what you're doing at this phase of the cellular life cycle is just copying what's in the nucleus, okay? After that, I'm gonna put a little break there, then you go through a second G spell. We call this one G2. And what's happening during G2, this is why they're called gap and not growth, because the cell's not really growing at this time. It's just kind of hanging out, but it is doing a couple things. First of all, it continues to have its normal cell function. So it's still functioning like a normal cell, but it's also preparing for division. So it might be making copies of organelles. It might be making enzymes that are needed during cell division, but that's what's going on during this G2 phase. And after it's hung out in G2 for a while and it's done all the preparations needed for cell division, then it's going to enter, and just draw it like this for me, guys. It's going to enter the M phase. The M here stands for mitosis. Along with mitosis, because mitosis, it turns out, is actually nuclear division. So we're talking about just the nucleus when we talk about mitosis, okay? So along with division of the nuclear material, y'all have seen pictures of cells dividing where the cell membrane starts to pinch in and eventually you end up with two different cells. That process of pinching in that physically divides the rest of the cell It also happens during this M phase, and it is called cytokinesis, which is a great name if you know your word roots. Cyto means cell, and kinesis means movement. So what's happening during cytokinesis is the cell membrane is moving in, and eventually it's going to pinch those two cells apart altogether, okay? Now, a couple things I want to point out about this diagram, okay? First of all, I told you that this mitosis piece is nuclear division. So that should clue you into the fact that what we are talking about here is exclusive to eukaryotes, right? This is not happening in bacteria or prokaryotic cells because they lack a nucleus. I do discuss in the lecture video how prokaryotic cells divide. That is a process called binary fission. It is super easy. I think I spend like 1 or 2 slides on it. I do expect you to know it, but it'll take you like 5 minutes to understand it, okay? So look that up. But this more complicated process only occurs in eukaryotes. That's the first thing that I want you guys to notice. The second thing that I want you guys to notice, and I'm going to switch slides, is that if you think back to your high school years, you may have been taught this acronym, IPMAT. Does this sound familiar to anybody? Were y'all taught this? I wish I could have got a hold of all y'all in high school. I would have taught y'all so many neat ways to remember stuff. Y'all be acing my class right now. This is how I taught it when I taught high school, when I taught like cell cycle and mitosis. It's easy peasy to remember IPMAT, and then once you know IPMAT, then you just remember what everything stands for. So y'all told me that you had heard the term interphase before. Yeah? That sound familiar to you guys? Okay, so could you define interphase for me? Like what is interphase? Based on your memory, like of 9th grade biology, which I know is asking you to reach back into the cobweb part of your brain. What you got? Well, that does happen during interphase, but what I was looking for is sort of like students come to me and they're like, interphase is just when the cell's chilling out, because that's what you're usually taught in high school. And it's not really wrong. It's just that for many years when we studied cells, the equipment that we used were microscopes. So we couldn't see what was happening a lot of times at the molecular level. So what you were taught in high school as interphase, which is the period when the cell is quote unquote chilling out, actually encompasses G1, S, and G2. So you're 100% correct. The DNA synthesis is going on during interphase. It's just that the interphase that you were taught in high school, and interphase is still a term we use, it encompasses these 3 phases together. And it is the time when the cell is performing its normal cellular functions and not actively dividing, okay? Does that make sense to everybody? It's just a combination phase. The rest of this stuff here refers to parts of mitosis, okay? And there are 4 phases to mitosis that I want you to know. Can anyone help me like remember what these phases might be based on these initials? Yep, I'm gonna write pro, meta, ana, and telo. Okay, so yeah, these are the phases of mitosis. Now, your book talks about a phase that's kind of a combination phase that it calls pro-metaphase. And I'm going to tell you why I hate that. It's like, oh, well, during pro-metaphase, it treats it like that's a distinct phase. None of these are distinct phases. This is a process. What we've done as biologists is said, well, when it reaches this point, we're going to call it this. And when it reaches this next point, we're going to call it this. So you could draw intermediate phases for any of these. Does that make sense to you guys? So I don't like to use this as a term, okay? I just, yeah, there's a point where it looks like it's halfway between prophase and metaphase because that's just how life is. It's a process. It's moving through these stages. Does that make sense to you guys? So if you see that in your book, you can kind of ignore it. I will talk about these individual phases. The other thing that I want you guys to know is that along with telophase, is when that cytokinesis piece is occurring. Now let's go through and let's look at each of these phases individually, okay? So we're gonna do some drawing, which means y'all have to put up with my fabulous artistic abilities. So let's look at it. If we were in a lab right now, and I was teaching you guys this by showing you cells in a microscope, let's diagram what an interphase cell would look like. So this is I'm keeping it real simple. Cell membrane, nucleus. Now inside the nucleus, what you would find is that the DNA would not be, it would be looking like kind of like a loose ball of yarn. And here's why. DNA in the eukaryotic cells, remember how I told you guys that DNA, if I were to take an individual strand of DNA from one of y'all's nuclei and I was just to pull it out and like walk backwards, you would have something like 6 feet of DNA in each of your individual cells, which is a ridiculous length for a molecule to be, right? So your cells have to organize this DNA in some way so that it doesn't start to look like a tangled ball of yarn that's not useful anymore, okay? So it turns out if you could zoom in on this DNA, what you would find is that it actually exists in a form where it's wound around a series of spools. And so it starts to look like a bead necklace. It looks something like this, okay? So this is basically the DNA right here. And the spools that the thread is being wound around are a series of proteins called histone proteins. And we're going to be talking about these again later in the course. So go ahead and learn them now, okay? This is actually a series. It turns out there's like 8 of them in here, but they basically make up a little donut like this that the DNA winds around, okay? This combination of DNA plus histones together is called chromatin. So in the interphase cell, what you can see is a darkened nucleus, and sometimes you can make out that it looks like a big ball of spaghetti because the chromatin you won't see any individual chromosomes yet, okay? The chromatin is kind of loose. It's like a bead necklace that someone just threw in the jewelry box, okay? It's called chromatin, so chroma means color. And if y'all have looked at cells like in our high school biology class and you notice that the nucleus is really darkly stained, well, that's where that came from. The nucleus takes on the stain really darkly, so that chromatin, that's where that name comes from, color, okay? So that's what an interphase cell looks like. Now, what happens when this cell starts to divide? Well, a couple things. First thing, there's our cell membrane. The first thing is if we want to divide up this DNA into 2 different cells, one over here and one over here, we got to get rid of that nuclear envelope, don't we? Otherwise, we can't access that DNA. So one of the first things that's going to happen is the nuclear envelope is going to dissolve. And so we'll just write nuke and dissolve or degrade, however you want to put that. Okay, it starts to go away. The second and really important thing that starts to happen is the chromatin condenses into chromosomes. Okay, so these histone beads start to stack on top of each other until they start to look like something y'all have seen before. So let's draw a chromosome really quickly. And I'm going to draw it a very particular way. So I'm going to draw that way. Hold on. Hang on. I'm so artistically challenged, y'all. Okay, so no, so I want this to be one piece, and you'll see why in a second. And I want that to be a second piece, and then I want y'all to draw like a button in the middle. Okay? The reason I drew it that way, so y'all have seen diagrams that are much better than this, but kind of like this in your textbooks before, right? Not if you're with me. So here's what you're really looking at. Remember how during S phase, the DNA was replicated? So you end up with 2 copies of all your DNA. All your linear chromosomes have 2 copies. This is one copy, and this is the other copy. Yes, I make my check marks backwards. I am left-handed. So, so yeah, this is one copy. This is the other copy. All right. So what you're looking at is a strand of chromatin that has where the beads have stacked on top of each other, and there's one of the copies and the other copies, and these two copies are held together by this button thing right here. Does anyone know what this button thing is called? Centromere. Yep. But hold on to centrioles because we fix and talk about them in about 30 seconds. Okay. So this is called a centromere. Okay. And you can think of it almost like a button or a staple that's holding these two copies together. Now, for each of these copies, we have a name for each of these legs, really is the way I like to think of them. We call them a chromatid. And together, these two are called sister chromatids. Okay? Does that all sound like familiar terminology you may have heard before? So what you'll start to see in this prophase cell is you'll start to see those familiar X's. So I'm just going to draw too big and too little here. We're going to assume this is a diploid beast, but we're not going to draw 46. We'll just draw too big and too little, okay? So you start to see these chromosomes condense out of the chromatin. The third thing has to do with the centrioles. So I'm going to write centrioles right here. So what the hell, oops, is a centriole? Come here, you. Okay, so draw yourself on either side. I'm going to blow it up here. I want you to draw something that looks like a little X made up of sort of like logs, okay? So I'm going to kind of draw it on here small if I can. You want one on either side. Actually, they're not on either side during prophase. They just start to form. Okay, so these centrioles, what they do, and that's what this is right here. This is a centriole. What they do is they make what are often called spindle fibers. Y'all have seen diagrams of cells dividing that look like Spider-Man has attached webs to the individual chromosomes. And the little web things are the spindle fibers. These spindle fibers are actually made up of cytoskeletal elements that y'all have heard of before. Do y'all remember the term microtubule? So these spindle fibers are microtubules. So what centrioles are is they make microtubules, is what they do. So you start to see centrioles appear and they start to make these spindle fibers during prophase, okay? So that's what's happening first. We're just getting the party started. It gets simpler from here, okay? Now let's look at metaphase. In metaphase, let's draw our cell. This is the cell membrane. Things to look for. At this point, our nuclear envelope should be gone or almost gone. Okay, so we've gotten rid of the nucleus, the envelopes. We can start dividing this stuff up, all right? The second thing that we're gonna notice is that our chromosomes are going to migrate to the middle of the cell. And here's why I say it like that. Metaphase, migrate, middle. This is how you can remember this. Check it out. So I said I had too big and too little, right? So I'm just going to draw them like that. Okay? So if you start to see these things lined up in the middle, you've got a metaphase cell. Now pay attention. Notice these guys are lined up single file. That's going to be important later because we're going to be differentiating meiosis and mitosis in a second, okay? So they've migrated to the middle. The next thing is your centrioles are at what we call the poles, and the spindle fibers attached to the centromere. So I'll put spindle fibers, and we'll put centromeres. So let's all draw that. Okay, so here's our little X's that make these microtubules. And they make these microtubules and they come along and they attach to that button in the middle, okay? So even though my drawing is terrible, this should look really familiar to drawings that y'all have seen before in textbooks, yeah? Okay. Now, here's the thing. What's going to happen, remember that each of these legs here, each of these chromatids is a separate copy of DNA. So where we're headed is this centromere can actually, unlike a button or unlike a staple, it can split in half, okay? So what's going to happen is these microtubules later are going to start to contract in opposite directions, and you're going to end up, yeah, like that may be a better analogy. I like that, Peyton. Think of the centromere as Velcro holding these things together, and then you've ripped them apart. That's excellent. And so where you're headed next is anaphase. And in anaphase, and I'm going to draw a little bit of a bulge, and you'll see why. In anaphase, that's where we start to see, I'll go ahead and draw my little centrioles. Okay, so we're going to see that these microtubules have started to contract and that the centromeres have split. So we'll draw that, and then I'll write all that out. So here's half a centromere, and I'm going to draw this with the little legs pulling behind because you got to imagine it's moving through some water, right? Moving through the cytoplasm. So the legs are dragging behind it. Does that make sense to you guys as to why it's drawn that way? And so same here. And then this one was supposed to be a little one, and that one was supposed to be a little one. Here, 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 here. Okay. So what we see in anaphase is the microtubules contract, microtubules or spindle fibers, whatever makes sense to you. And then you should see centromeres break. Okay. Now, I told you you could remember metaphase because the chromosomes migrate to the middle. Here's my trick for remembering anaphase. If you turn your head and you made a little bar across this, doesn't this look like the letter A? You see what I'm saying? Like a capital A. If I drew like a line here, that's an A, right? Sort of. I don't know, it's a stretch. Hey, don't make faces. It's a stretch, but you see where it's, okay, are you with me? You believe me? You made a face. So I feel you. So I was all about like ways to remember this process when I was learning it. And so to me, I'm like, oh, okay, anaphase, letter A, this kind of, the chromosomes look kind of like a sideways A. Maybe that's not, that should be sideways A, question mark. That, if that's not helpful to you, don't use it, but that's how I remember it, okay? So we've done interphase, prophase, metaphase, anaphase. What's left? Yes, let's do that. Telophase. Okay, so for telophase, I'm going to actually draw something that looks like this. These are our cell membranes. Now, this part here, remember, that's not technically part of mitosis. That is the result of cytokinesis. So cytokinesis is going to eventually pinch that cell membrane off, right? But this is often happening at the same time or right after telophase, so I like to draw them together, okay? Now, at this point, what you should start to see is the development of two new nuclei, okay? So you're going to start to see regeneration. So I'm just going to put regen of the nuclear envelope. Okay. The next thing that you're going to see is, and I'll draw one this way. So now at this point, we should have like little individual pieces of our chromosomes, right? Because if I go back here, you can see that this is just like one leg, one leg, one leg, one leg, right? One leg, one leg, one leg, one leg. But what happens very quickly after they separate is the truth is they start to decondense back into chromatin, okay? So this is what's going on, but this is what it starts to look like. So chromosomes, I'm just going to put chromes, relax into chromatin. Okay? And then, of course, the last thing that you would have happen is the cell membrane divides. So notice I haven't said anything about replicating organelles or anything. Sometimes some of that might happen like during G2. Some of that happens after cell division during G1 because you now have the instructions for how to do all that, right? The end result of mitosis is two identical diploid cells. Do y'all see that? Do you believe me? Because that's going to be really important because we're going to move on to meiosis here in a second, okay? All right, let's compare this with meiosis, all right? So in meiosis, we are talking about formation of gametes. So those are your eggs, those are your sperm. So this is obviously related to sexual reproduction. And just so that I know that we're all on the same page, can someone just give me like a layperson's definition of sexual versus asexual reproduction? What you got?
Asexual, you don't really have to have a partner.
Yep.
You just produce on yourself. You produce by yourself.
Yep.
And the other one
Okay, so she said reproduce by yourself if it's asexual, and you have a partner if it's sexual. And in terms of the genetic makeup of the offspring, in asexual reproduction, genetically, what are the offspring like? They're identical to the parents, that's right. In sexual reproduction, I'm trying to make sure I'm distinct in how I say that. When you're talking about sexual reproduction, how do the offspring look relative to the parents in terms of their genes? Mixed, yeah. So, and really unique because, like, for example, yeah, I got half my genes from my mom and half my genes from my dad, but which ones I got from mom and which ones I got from dad are random. So if you were to see me next to my mom and dad, you might think I look a little bit like both of them, but I don't look exactly like either of them, really. Okay? So let's talk about how we're going to do that. So I told you guys, just to remind you where we're heading, if you're talking about a regular cell that's got 46, what we're going to end up with is 4 cells that are at 23 each. And I've made those too small, but y'all know what I'm talking about. So y'all have heard of 23andMe, like when you get your DNA sequence, that's what they're referring to, is the 23 different kinds of chromosomes. And I think that's why students get confused. They're like, yeah, we have 23 chromosomes. You do, you just have 2 copies of each, okay? So let's think about, okay, in order for us to understand a little bit about meiosis, we need to sort of take a look at maternal and paternal chromosomes. So I'm going to do this very sort of gender normative thing, and I'm going to make mom's chromosome pink just for ease of explanation and dad's chromosome blue. Which incidentally, there was a whole article I read about how blue used to be associated with girls. There's some, yeah, I can't remember now, but it's just a stupid marketing thing, but I just want you guys to be able to tell the difference, okay? So, in looking at these 2 chromosomes, things that I want you guys to be aware of. So these are 2 copies of the same chromosome in this example that I've drawn for you. Can you see that they're supposed to be, even though I'm a terrible artist, the same size and the same shape? And if I was a better drawer, the centromere would be in the same place. Can you tell that I'm trying to draw 2 identical chromosomes? You got one from mom, one from dad. But let me be careful about what we mean when we say identical. What we mean when we say that they're identical is that they have the same genes. Okay, now they could have different versions of each gene. And those different versions are called alleles. Now, what the hell do I mean by that? Okay, so like, let's just say that mom had brown hair, okay? But dad had blonde hair. Can you see that the gene for hair color is what's on this chromosome? The version that mom had was brown, the version that dad had was blonde. But this is still considered two identical chromosomes because they both contain the gene for hair color. Y'all with me? Okay. We have a name for these identical pairs of chromosomes, and they are called homologs. Or you may see it just written as homologous pairs. And this name should make some good sense to you because you guys know that the prefix homo means same, right? So now one thing to note, first of all, people confuse homologs and chromatids all the time. So like, can you see that these are the two sister chromatids of this chromosome? These are the two sister chromatids of this chromosome. These two together are a homologous pair. Another thing, as we go through meiosis, that I want you to be aware of is how to figure out how many chromosomes you have at any point in time. Because it's important to know whether you're at a diploid situation or a haploid situation. And I'll tell you now that when you're interpreting diagrams, when you're looking at meiosis, the way to figure out, well, meiosis or mitosis, the way to figure out how many chromosomes you have is to count centromeres. And just put a pin in that, and I'm going to show you what I mean in just a second. Okay, so just kind of remember, I told you to count centromeres, and I'll show you why you need that. All right, now, in thinking about how meiosis is going to accomplish this task of going from, let me go back, meiosis here is going to accomplish this task of going from 46 to 23, or if you were a goldfish, they have like 96 chromosomes, so whatever half of 96 is it 48? Yeah. Anyway, so yeah, whatever species you are, if you're diploid, you have twice as many, right? And if you undergo meiosis, you're dividing that number in half. So meiosis is often called a reductional division. So let's do a real quick meiosis overview. All right, so this is just kind of an overview of meiosis, and then we're going to get into the specifics, okay? So I told y'all that you're going to start out with a diploid organism, and so we're going to use, I'm actually just going to draw some little chromosomes here. Let's make this a little bit bigger. And in this example, I'm going to say that we have, like before, two big and two little. And I'm going to have this be mom and this be dad and this be mom and this be dad, okay? So the first thing that's going to happen in meiosis is we're going to actually attach microtubules to the centromeres. Ooh. We're going to attach microtubules to the centromeres, and we're going to pull in opposite directions like we did in mitosis, but here's where the difference is. The centromeres are not going to split. So you end up with one big and one little in each of these guys. Do y'all see that? You see how what we did is we pulled them apart, but we didn't unstaple the papers, right? Does that make sense to you guys? So this is called meiosis 1. Okay. Now the next thing is, remember that we have an X here still, right? That means we've got 2 copies. So we don't need 2 copies. We need to unstaple the copies from one another now, okay? So now we're going to undergo a second division. And in this circumstance, we're going to pull in opposite directions. But we're going to split the centromere this time so that you end up with 4 resulting cells, each with one big and one little, but just one copy. Now, this is why I told y'all to count centromeres. This is, by the way, this is called meiosis 2. Easy peasy, right? Now, which of these, meiosis 1 or meiosis 2, is the reductional division where you go from being 2N or diploid to N or haploid? You say 2? Who else says 2? 2? 2? I tricked you because I knew you were going to say that, and it's wrong. And this is why it's important to count your centromeres, okay? This is why my handy dandy tip of counting centromeres is so important. Let's count centromeres. 1, 2, 3, 4. 4 chromosomes here. 1, 2. 2 chromosomes here. You got 2 copies of that chromosome, right? But it's still, you got like 1, you see that? Do y'all see what I'm saying? Now, if we unstaple these guys, look, 1, 2, 2, 1, 2, 2. And do it again here. So all of these guys have 2 chromosomes. Do y'all see that? Now that you've counted centromeres, do y'all want to change your answer? Yeah, meiosis 1 is considered the reducing part. Okay? Questions about that? I know that can be a little confusing, but does it make sense after you counted the centromeres? And do y'all see how this is just 2 chromosomes? Yeah, they're duplicated, but it's just 2. And then once you pull them apart, you have 1 copy of them in each there. Might have been easier if I'd shown you like, this is mom's chromosome, this is dad's chromosome. Now you just have moms, but you pulled apart the copies of moms. Before you had moms and dads. Does that make sense? Okay, let's get a little more specific. Hold on, I need to give myself a couple more slides because I know I'm going to run out of space. Okay, okay. So back to this Racha. Let's look at what happens during meiosis. Meiosis 1 is really the one that's hardest because it has a few unique features. If you understand mitosis, it's pretty easy to understand meiosis. But let me give you a little tip, things that you're going to need to know. I promise that there will be questions on the exam asking about the differences between meiosis and mitosis. Some of those questions will take the form of visual, like I may have some diagrams, and then you'll have to say, this is, metaphase 1 of meiosis. This is mitosis. And you need to figure out how you can tell, okay? And there are ways to tell. So let's do that. All right, I'm gonna assume y'all know what an interphase cell looks like because it looks the same for meiosis or mitosis. If we're talking about prophase 1 of meiosis, here's what we're looking at. So you have your cell, and just like before, we're going to need that nuclear membrane to break down. We're going to start making we're going to have our little centrioles here so we can start making our microtubules. Our chromosomes are going to start to condense, okay? But a couple things I want to point out. Okay, so just like in the other one, you're going to have all the same things happen here that you saw happen in mitosis. Nuclear envelope's going to degrade. Your chromosomes are going to start to condense. Here's some unique stuff that happens. Unique stuff that happens is that the homologous pairs match each other. And I'm going to put match in quotes. What I mean is they kind of line up. Y'all see how I drew these guys side by side? And I drew these guys side by side here. I did that on purpose, okay? This idea of the homologous chromosomes finding and lining up next to one another, there is a name for that, and that is called synapsis. Now, the second thing that can happen at this point is these things actually get so close to one another that their legs can become tangled up. And then when they become untangled, they can swap pieces of chromosomes, okay? That is a process called crossing over, okay? Why is that important? Well, let me go back a couple slides here. Go back to this one. Let's just say that these 2 chromosomes got tangled up during synapsis and that this leg here overlapped with this leg here. And when you untangled them, they swapped. Well, they would both still have the gene for eye color, but now suddenly this one would have the gene for brown color, and this one would have the gene for blonde color. Or no, I said eye color, I meant hair color, whatever my example was. What was the example I used? Hair color. Okay, I think I had blonde, brown hair, right? Now, that means that now you kind of changed up those chromosomes a little bit, right? So now whatever else was on dad's chromosome has been mixed up with some of mom's traits. Does that make sense? So crossing over is going to be an important way that we generate genetic diversity, okay? We're gonna talk about genetic diversity in just a second. All right, so that's some important stuff that happens during prophase 1 of meiosis. Let's talk about metaphase 1. All right, so if I gave you a diagram and it was prophase 1 of meiosis versus prophase of mitosis, the way you would tell is you would look for whether or not the homologs were paired up. You might even see the legs all tangled up, okay? Now let's look at metaphase. Remember that these homologs are paired up, so they're gonna line up together. They're gonna migrate to the middle, just like before, but they're gonna line up together. And then we'll have our spindle fibers here and here, spindle fibers here and here. So, there's a little X, okay? There's our centriole. So, this looks really similar. The chromosomes have migrated to the middle, haven't they? The microtubules have attached to the centromere, but here's the difference. The chromosomes line up, and I always say 2 by 2. Does that make sense? Let's go back up here. Let's look at how it looked up here. Do y'all see how that was single file? And it was single file so that you could pull the Velcro apart, right, Peyton? Here, they're 2 by 2. Because what we're going to do is pull the entire chromosome over to each side. Does that make sense to you guys? So if I showed you this picture or one like it on the exam, and I would ask you to say, what is this, meiosis or mitosis, and which phase? And you would have to be like, that is meiosis 1 metaphase, okay? Or metaphase 1 of meiosis, this might be how you'd say that. Okay, does that make sense? 2 by 2 is how you would tell. All right. Now, let's look at anaphase 1. It's going to look similar to what you've learned before as well, except for, so I'm going to have my little spindle fibers here. I'm not going to draw the centrioles because I'm going to be lazy. Here's a centromere, here's a centromere, here's a centromere, here's a centromere, but look, you're pulling over the whole thing. So can you see that I've got both chromatids going? Can you see that? So again, if I showed you just this picture, you should be able to tell me, yes ma'am, hold on one second, that this is anaphase 1 of meiosis because we're pulling the whole thing over. Now tell me what your question is. That's exactly right. That is exactly right. We are not going to break the centromeres yet. That's next. So that is, you have gotten to the crux of the issue immediately. So yeah, you're not breaking those centromeres, you're pulling the whole damn thing over, because when we get to telophase 1, what you're going to end up with is 2 cells. Now, I'm gonna ask you a question here. And before you answer it, I want us to go back to our OG up here. One of these chromosomes is from mom, and one of these chromosomes is from dad. One of these is from mom, one of these is from dad. Mom, dad, mom, dad. Mom, dad, mom, dad. Are these genetically identical cells? No, they are not, because even though they have the same genes, they don't have the same versions of those genes, right? We all have the same genes. We all have genes that tell our bodies how to make a liver and grow a thyroid and whatever. We don't have the same versions, and that's why we look different. So these are not genetically identical, okay? Okay, now, let's go to meiosis 2. So we started here. Now, you may be like, okay, is it gonna like decondense and go back to an interphase? It really depends on the process of gamete generation for the organism that you're talking about, okay? So some will just go straight into their next division, and these won't be necessarily lined up in the middle, okay? But sometimes you won't have another interphase. It'll just go straight into prophase 2. Prophase 2 is kind of where they're condensed already. And so I'm gonna pick up at metaphase 2, okay? So in metaphase 2, what we've got at this point is, and I'm going to put an M up here for mom's chromosome and mom's chromosome and dad's chromosome and dad's chromosome, right? So metaphase 2, what's going to happen? And this would be hard. I'd have to give you a lot of information for you to be able to tell this from mitosis, wouldn't I? I'd have to really give you some information, and I would make it very clear if I expected you to know the difference, because this is single file and that's single file. And so just like before, what's going to happen is we're going to have microtubules attached to the centromeres. And then what's going to happen in anaphase 2 is, sorry, I'm doing a terrible job drawing this. It's elongated. They look like onions or something. Okay, but you can see that what's going to happen is the centromeres are going to split now. Now they're going to split down the middle, and you're going to end up with one half going that direction and the other half going that direction. There's a little centromere, 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 until you end up with a situation like that. And I'm not going to draw the other one in the interest of time, but you see how you end up with 4 cells. So you have to divide twice to get to that 4. Okay? Questions? It's not hard. It's really not. It's just not obvious right away. If you guys draw this like 2 or 3 times to study it for your exam, I think you'll be in really good shape. But to me, the only way, like people ask me all the time, what are your study tips? Like sit down and draw this, and then draw it the first time using my notes and your notes, and then see if you can reason your way through it without looking at anything for help, okay? If you can draw this stuff from soup to nuts with no help, you're in good shape, okay? So we're getting close on time. Let me make you a list of the things that are in my lectures, but I didn't have time to talk about today. Okay, what I gave you was the fundamentals of meiosis and mitosis so that you can go back to those lectures and rock them and understand everything. In chapter 12, now of course, everything that's in my lecture, I expect you to know. Things that I think you guys should really hone in on, okay? First of all, everything I told you about cell cycle and cell division today was about eukaryotes. I do want you to know a little bit about how prokaryotes do things. That is a process called binary fusion. Okay, binary meaning 2, fusion, divide. So make sure that you look that up. Then the next part of chapter 12 that I want you to pay attention to is how the cell cycle is controlled. And I'll tell you right now, a lot of it has to do with 2 different molecules, one called cyclin and one called CDK, which stands for cyclin dependent kinase. And the reason this is important is because if your cell cycle is not properly controlled, either the cell can die, or what happens if the cell cycle is not controlled and the cell just divides and divides and divides and divides? What's that called? That is called cancer. So if the cell cycle is dysregulated, you get cancer. Cancer at its, now all cancers are different, okay? Prostate cancer is different than breast cancer because they have different ways they develop and different ways they spread, and some of them are more aggressive because they, have all these different characteristics. But at their core, the basic problem with all cancers is that the cell population that the cancer came from stopped understanding when to stop dividing. Okay, some of you guys may know my husband was recently diagnosed with cancer. So if I seem very off, that's why. And it's because his cells, his prostate cells just stopped figuring out how to stop dividing. And then once that happens, they divide and divide, and they start dividing so quickly. The reason that cancer is so dangerous is when they start dividing really quickly, they stop taking the time to characterize themselves as the tissue type they came from. In other words, like when my husband has prostate cancer, so his prostate cells divide and divide. They don't take the time to differentiate into prostate cells. They just look like prototypical cells. So they can leave the prostate and set up shop anywhere in your body and then start adding tumors other places. Has anyone heard of metastatic cancer before? That's what a metastasis is. It's when you have these little individual cells that no longer look like the tissue they came from, and they start seeding other places of the body, okay? So I talk about that in chapter 12. All right, chapter 13, things that y'all need to know about. Let's see here. I had a list of stuff. Where did I put it? Oh, okay, I know. So the big thing in chapter 13 that I didn't get to talk about that I wanted to talk about for you guys is, first of all, how does meiosis contribute to genetic diversity? So I think most of you guys probably know that one of the reasons that sexual reproduction is so prevalent throughout our biological world is because it generates genetic diversity. And most of you understand that genetic diversity is really important to the health of a population. COVID-19 is an excellent example of that. If all of us were equally susceptible to COVID-19, we would all be dead, right? But some of us are asymptomatic. Some of us, like me, I felt terrible for a couple weeks, but then my only lingering effect was I couldn't taste for a while, you know? And then there's some people that end up in the hospital. And a lot of that has to do with our differing genetic makeup. But in addition to the fact that you have different individuals that are fusing and that generates genetic diversity, there are some ways that meiosis generates genetic diversity. And so let's kind of, 3 that I want you to pay attention to. Obviously, the act of fertilization, which is related to meiosis because meiosis is what makes the gametes that are going to be fusing during fertilization. I want you to understand how crossing over contributes to genetic diversity. And then the last thing that I explain and talk about in the video that you need to know for your exam, because I will ask about it, is independent assortment, okay? And that's defined in the video, and you'll see how that contributes to genetic diversity. And then the last thing in chapter 13 that I didn't get a chance to talk about today that I want you to look up is how things go wrong. If you have a mistake in meiosis, you generate a faulty gamete. If you generate a faulty gamete, then you can end up with some offspring that have problems. One very common example of a disease that we see in human populations that's the direct result of a mistake during meiosis is what? Anybody know? Down syndrome. Down syndrome is the classic example. There are others, but that's the one, okay? So make sure that you guys look at that stuff. I will try to post some of those discussion questions like I normally do. It won't be today because storms and things like that, probably tomorrow. Also, don't forget that you have homework. Only 1 chapter's worth this time, but I'm still giving you full credit like it's 2 chapters worth. So you're welcome. Study, okay? All right, I'll see you guys next week.